The title of my presentation is Lipidomics and Metabolomics-based stratification of COVID-19 patients. Thank you for your kind presentation, and I will introduce our work. So, our work is the summary of our last published paper on scientific reports, dysregulation of lipid metabolism and pathological inflammation in patients with COVID-19. This work was published in the February of 2021, but we have performed all the experiments at least 6 months earlier. And I want to acknowledge my institute, which is the Change Advanced Biotechnology, because we were able to build a COVID task force with fundings from the Campania region, which is the region in which Naples is located. I highlighted 2 of my lab members, which is Dr. Marianna Caterino and Professor Margherita Ruoppolo, and this work is the sum of all the efforts of other people from other groups, and it is a collaborative project to introduce the aim of this study. We wanted to identify the pathways and the processes that could be involved in the disease progression, just to have the chance to classify the disease according to several severity degrees, and also to identify new therapeutic strategies. The question that we want to give an answer to is, how does the metabolome and the lipidome change according to the severity of the disease? So we know that small molecules, the metabolome, is involved in several functions of the immune system. For example, amino acid and amino acid related, such as tryptophan, BCAA, or kynurenine, are involved in these functions. But also lipids are main mediators of the inflammatory response, such as the ceramides or the cell signaling, for example, the acylglycerols, triacylglycerols, and the phospholipids, and also acylcarnitines, which are involved in the mitochondrial dynamics for energetic homeostasis. What we are going to see are the results from a 53 patients cohort of COVID-19 patients. So these patients were diagnosed with molecular testing, and in particular, we differentiated 3 different groups: mild patients, moderate patients, and severe patients. From these patients, serum was collected and analyzed by targeted metabolomics. Then data analysis followed for statistical analysis and data visualization. Finally, a correlation of the metabolic changes with the inflammatory cytokines was performed. So let's start with the presentation of the patients. As I told you, 53 patients were enrolled, and we had the proportion of 70% males and 30% females, with a median age of 58 years. So patients were classified using a 7-point scale. For example, I can show you the first 3 points that we have used to characterize mild patients. So patients for the point 1 and point 2, not hospitalized, but patients able to command their life and do their normal activities, showing only mild symptoms. And for the point 3, hospitalized patients, but that not required oxygen for the respiration. I want to punctualize that for this study, we have only used patients according to the point 3, just to exclude external influences from the normal lives of these patients. Then the second group is the group of moderate patients that are patients hospitalized, but that required supplementation of oxygen. And finally, the third group of severe patients, in which all the patients were hospitalized, required supplemental oxygen, and also non-invasive or invasive mechanical ventilation. Unfortunately, some patients died for the disease. Then for the methodology, our clinicians collected serum samples at the hospital and stored them at -80 until our analysis. We only use 10 microliters of serum from each sample to perform a targeted metabolomics preparation and subsequent mass spectrometry analysis according to the protocols provided from Biocrates for the MXP 500 kit. And finally, we prepared 3 independent samples from the same patient, from each patient, to be used as a technical replicate and injected and analyzed by LC-MS/MS or via MS/MS. So using the instructions, we decided to focus on the lipidome of this patient's sera. In particular, we went able to identify and quantify 6 classes of lipids for a total of 483 molecules, which included diacylglycerols and triacylglycerols, which were 44 and 242 molecules, glycerophospholipids in the sum 19 molecules, cholesterol esters, ceramides, and sphingomyelins. For our experimental setup, we have conducted all the MS analysis on a triple quadrupolar Q-trap system coupled with an Agilent HPLC chromatograph. And finally, the data that were acquired using the software from Biocrates were analyzed for multivariate statistical analysis using metabolomics 5.0 software. In particular, we performed the imputation of missing values to eliminate the 0 values on concentration values. Then these values were log2 transformed and finally scaled according to Pareto scaling method. Then we have performed univariate statistical analysis and correlation analysis using GraphPad Prism 8 version. For the multivariate statistical analysis, I can show you the results from partial least square discriminant analysis, which was able to show the variance between the 3 analyzed groups. So as I told you before, mild, moderate, and severe groups, which separated according to the principal components. And also from the same analysis, 28 molecules identified as the most discriminant lipids molecule according to the VIP score, which is the variable importance in projection, setting a threshold of 2. Using the most discriminant molecules, we could identify 12 molecules which increase, as you can see, we have a legend here, from the 0 to 2, which is mild, moderate, and severe groups, following a pattern of variation in the abundance, which range from the blue, which is the low abundance, to red, which is the high. We followed this pattern and we could identify 12 molecules. The same way, we identified other 12 molecules which decreased in the severe group respect to the moderate and respect to the mild one. In particular, we can see this pattern, which is red for the most abundant, then we have medium value, and the lowest value, which is represented by the blue color. We performed clustering and heat maps visualization and separated all the data according to the lipid class. So we have glycerophospholipids, triacylglycerols, diacylglycerols, cholesterol esters, ceramides, and sphingolipids. As we can see from all these heat maps, there is a very huge alteration in the lipid homeostasis induced by SARS-CoV-2 infection, because we have a lot of patterns in all these lipid classes. And finally, we performed the univariate statistical analysis, calculating the statistical difference in the lipid concentration, applying parametric or non-parametric tests according to the distributions of our data. In particular, we could identify the differentially abundant lipids according to a p-value of lower than 0.05, and according to a log2 fold change higher than plus 0.58 and lower than minus 0.58, which corresponds to a fold change of 1.5. Then, applying these two criteria, we have performed two comparisons, which means severe versus mild patients and severe versus moderate ones. So for the severe versus mild, as you can see in the volcano plot, we could identify, represented by the purple dots, 28 molecules which are differentially abundant in severe versus mild. In particular, 16 were more abundant and 12 were less abundant in severe COVID patients respect to mild ones. Then I have plotted all these data showing the abundances, so log2 fold change, in order to visualize the increase and the decrease of specified molecules. In particular, we noticed that there was a pattern of increased abundance linked to ceramides molecules and a decrease in the levels of exocyl ceramides. And intersecting this data from the data obtained from the multivariate statistics, we could find several commonly lipids with the same trend of dysregulation, which means upregulated for the up arrow and downregulated for the down arrow, which showed the same trend as found from the univariate statistics. And also in this case, we noticed that we had upregulation of ceramides and downregulations of the same exocyl ceramide. We did the same for the comparison of severe patients versus moderate, and we obtained a lower number of differentially abundant lipids. In particular, 18 that were 9 up and 9 down represented in severe patients. So, as previously, we have the graph related to the abundance of these lipids, and we found a ceramide which resulted upregulated and an exocyl ceramide which was downregulated. And we also intersected this data with the multivariate statistics, and we only found a diacylglycerol molecule as a commonly alterated lipid, and not for the other molecule. Then we decided to find a correlation, or to test if there was a correlation with the disease progression and the levels of proinflammatory cytokines, which we know that are commonly produced in the cytokine storm and in the pathology of COVID-19. So we measured the levels of the thymic stromal lymphopoietin, which is a cytokine produced mainly in inflammatory disease and in allergic disease. And we analyzed the levels of these TSLP in a T0 population and a T1 population, which means 22 patients at time 0 during hospital admission and after 7 days. We found no statistical difference between point T1 and point T0, despite the levels of TSLP after 7 days resulted similar or increased in the majority of the patients, except for one patient in which we observed the opposite trend. But when we clustered the concentrations of TSLP according to the severity of the patients, we found no differences, no correlation of this cytokine with the severity of the disease at time 0, but we found a correlation with the disease progression, which means that the levels of TSLP were higher in severe versus moderate and in severe versus mild comparisons at time T1, which means 7 days after hospitalization. So this showed that there was an association between the severity of the disease and the levels of this cytokine. But we tested the serum levels of many other pro-inflammatory cytokines, TNF-alpha, interleukin-6, interleukin-17, interleukin-26, and adiponectin, and we found no correlation with the severity of disease and increment the augmentation of these cytokines. Then we decided to test the correlation of within the lipid changes and the cytokines incrementation. And we correlated the total number of the quantified lipid, which was 483, with the above mentioned cytokines. And one interesting result was that we found a correlation with a group of lipids which were exactly the ceramides, which were the molecules that we found increased in the comparisons of severe versus mild and moderate patients. In particular, there was a positive correlation with interleukin-6 and interleukin-17, and a negative correlation just for one ceramide for TNF-alpha. In particular, we are talking about 18 carbons atoms. We don't know why this increase in ceramide and why there is a correlation with the inflammatory state which is increased in severe patients. We know that ceramides are certainly involved in this inflammatory state, but of course we want to try to understand if there is a causal type relationship between the increment of these lipids and this respiratory distress, the pathological inflammation, and the cytokine storm, which is characterizing COVID-19 patients. And we are trying with our future investigations to focus directly on this network, which is quite intricate, and trying to understand which is the cause of the increase of ceramides. We have an hypothesis or just a suggestion looking that in the pathway of ceramide there is serine, which is an amino acid that we have dosed in our metabolomics data set. In fact, I can anticipate just this datum because we have not published the paper yet, and we have a statistical difference between serine levels in severe patients, which are increased with respect to moderate, and in turn increased respect to mild ones. And as last information, I want to give you this hint that a patent was deposited from my institute and our PI. In particular, you cannot understand, I know, because it's the Italian title of the patent, but this is endogenous ceramides and exocyl ceramides used as biomarkers to discriminate the severity of SARS-CoV-2 infection, coronavirus infection. So I don't want to in-depth on this fact, but it's just an information that I want to give you, that a patent. So these molecules, in particular the 18 atoms, carbon atoms, can be used to discriminate the severity of COVID-19 disease. So just a brief summary, we have performed the full characterization of the lipidome of these COVID-19 patients' sera, and these patients have been divided in 3 subgroups according to the severity of the disease. There is a correlation with some inflammatory cytokines, and in particular between the severity of this disease and the cytokines, and a correlation between the ceramides and the cytokines. and this can constitute a predictive model that can link the dysregulation of the lipidome to with the stage, the clarification of the stage, the outcome and the progression of the disease in COVID-19 patients. Our future work will provide a new characterization based on a larger cohort. We know that our cohort is not the largest possible, but we performed these experiments quite immediately after the beginning of the pandemic, so we could not access a lot of patients, but we promised to repeat our experiments, enlarging the cohort and including, of course, other characteristics from the patient. Then we want to absolutely integrate lipidomics data, our lipidomics data, with the new produced metabolomics data, of course, highlighting the network of ceramides, and also integrate lipidomics and metabolomics data with other proteomics experiments. Our group has also an expertise in proteomics, and so we are performing some other experiments to validate our lipidomics and metabolomics data. Of course, our future attention will completely be placed on the ceramide network, because we believe in this network and we want to give a definite role for these molecules. So I want to show you a picture of some of the components of my research group. This is me, this is Dr. Marianna Caterino and Professor Margherita Ruoppolo. I just want to acknowledge my institute and my university department of my university, and of course, thank you for your kind attention.